So let's talk about measuring population size. So typically, it's not possible in terms of time, or it's not cost effective to count each member of a large population. So scientists can use different ways to estimate the size of a population. The two most important types of sampling used in ecology are the Quadrant Method and the Recapture method. So in the Quadrant Method, scientists are going to block off small areas of the habitat, and they're going to count all the organisms in that quadrant. They're going to do that with multiple quadrants. They're going to take the area of that quadrant and then use that to estimate how many organisms are in the whole habitat. So this type of method works best for either organisms that are immobile or things that are slow moving, maybe like a slug or something. Another method is called the Recapture method. So this works best for things that are moving, mobile organisms. So it involves taking a sample of organisms and then somehow marking them either with a tag or paint or a band or some kind of transmitter. The marked organisms are then released back into their original environment, and then sometimes later, a new sample is collected from the same environment. So that new sample should include some of the marked individuals from the first sample and some individuals that were never caught or captured during that first sample. The ratio of the marked to unmarked individuals can then be used to estimate the entire population size. So let's look at a sample and a formula for how to do that. So here's the common formula we're going to use. So we're going to take the ratio of the marked individuals over the total population size, and we're going to set that equal to the number of organisms that are recaptured in the second sample divided by the size of the second sample. So we're trying to find the population size, the total population size, N. So let's solve our ratio for N to get this equation. So N is equal to the total of marked individuals in the first sample times the size of the second sample, lowercase n, divided by the number of organisms recaptured in the second sample. In that case, this is M. So let's use this in a sample problem. So here's our sample problem. An ecologist samples and marks 20 birds from a population. So we're going to be looking for N. So again, we're looking for N. So what we just found out is that we're taking a sample of 20 birds. So that's going to be the, well, let's hold that for a second. We're going to mark 20 birds. So that's the total of marked individuals. That's capital M. So N is going to be equal to 20. Keep reading. 2 weeks later, he captures another sample of 20 birds. So the second sample size is also 20. And that's N, little n. Keep reading. In the recaptured sample, there are 5 birds that were marked from the first sample. What is the estimated size of the bird population? So 5 of the original 20 were recaptured in the second sample. That 5 represents little m, the number of recaptures. That goes on the bottom of our fraction. So to estimate the sample, the population size, 20 times 20, 400, divided by 5, that's going to give us an estimated population size of somewhere around 80. All right, so that's using the Recapture method. And again, what we're trying to do there is estimate the population size.